Thank you and good evening. Welcome to ESF, to the Travis Lecture. My name is Christopher Nomura, and I'm the Vice President for Research here at SUNY ESF. And so I'd like to welcome you all in coming to this prestigious lecture. I'd like to give you a little bit of background about Dale Travis, who has been an incredibly generous sponsor for this program. So since 2013, because of his philanthropy, we've been able to have our stellar researchers here at ESF be able to deliver and express their research content to you, the public, in this forum. And it's been wildly successful. We've had great experiences and great topics over the years, and we have another great one today. So what I'd like all of you to do is put your hand up in the air and wave and say, "Hi, Dale." Okay, unfortunately, because of the weather conditions in New York City, Dale and his family were unable to join us today. But we know that they're with us in spirit and, of course, in their support of this particular program. So I'd like you all to put your hands together and thank the Travises for their generous donations to help make this possible. Thank you. So it gives me great pleasure to introduce today's Travis Lecture speaker, Dr. John Farrell. Dr. Farrell is a professor of aquatic and fisheries science in the Department of Environmental Forest Biology here at ESF, and he also serves as the director of the Thousand Islands Biological Station, also known as TIBS. John received his associate degree at the Finger Lake Community College and his BS degree at Cornell University in the Department of Natural Resources. He then saw the light and came here to ESF to continue his graduate work, where he completed his Master's of Science and PhD degree under Dr. Bob Werner while working at the St. Lawrence River. He currently teaches aquatic and ecosystems restoration and management, and he also teaches the Capstone Senior Synthesis class in aquatic and fishery sciences. He's also co-taught Adirondack fishes at the Cranberry Lake Biological Station for many years, in addition to a variety of other courses in the Department of Biology. John has worked on the IJC Environmental Technical Working Group and serves on the advisory boards of the Cornell Biological Field Station and the Central Michigan Biological Station, and he became a Roosevelt Wildlife Station scientist in residence in 2013. He has also served as an associate editor for the journal Wetlands and as a guest editor for the Journal of Great Lakes Research and Hydrobiologia, and has recently co-edited a book with his former students on muskellunge management published by the American Fishery Society in 2017. Now, I could go on with a number of accolades for John, but I want to let him get to his talk this evening so we can hear his really interesting research. So please join me in welcoming our speaker today, Dr. John Farrell.
Thank you so much for everybody coming in. I really appreciate it. I'm very thankful that we didn't have a lake effect snow event or something today. We've been worried about that, but it's wonderful to see so many familiar faces and many new faces with interest with the issues that we face here on the St. Lawrence River and the Great Lakes that I'm going to focus on. So the big fish in the balance you probably saw is the muskellunge that we're going to be focusing on today, but it tells a story of how conservation of this fish has evolved over time between ESF and many of its partners, and the gift of an island to the college that merged into a diverse aquatic and research program that exists in the Thousand Islands on the St. Lawrence River. And so I hope to achieve this difficult task of telling this story, and I hope you enjoy it. So just as an overview, so you can kind of understand where it's going, I want to give a short introduction to the St. Lawrence River and talk about some of the environmental and ecological changes that have occurred to this massive ecosystem. And then talk a little bit about our mission at Tibbs and what we do and how our research and management model works. And also focus on the research and how it targets restoration and sustainable use of aquatic resources, because that's really what we're all about. And I want to make sure that I give thanks and some information on the collaborations and student research that we focus on there. And I've spent a lot of time on acknowledgements because so many people have helped to make this happen and help propel Tibbs in my career. And also talk about the history and the development of an ESF field station. So that's what I hope to achieve with the talk. So getting right into it, the St. Lawrence River is one of the largest freshwater ecosystems on the globe. We have a drainage basin, which you can see here in this tan coloration, that's over 1 million square kilometers. So it's one of the principal drainages of the North American continent, and it drains the entire Great Lakes ecosystem. The natural outlet is through the St. Lawrence River right here in upstate New York. You can get there in about an hour and 15 minutes from where we stand right now. So we have this exceptional resource that's the drainage of the largest freshwater, surface water resource on the globe. So it's quite an enormous task to manage these things, and we have, it's also an international waterway shared between the US and Canada. And we have a Great Lakes Fisheries Commission and an International Joint Commission that help manage these water and resources jointly through treaties that have been founded. So I got to go back. So in the upper St. Lawrence River, there's an archipelago of over 1,800 islands in the Thousand Islands section of the upper river. And it's just a remarkable ecosystem. And you have this deep water environment that goes through the seaway. There's places over 70 meters water depth, over 240 feet deep. And they're right adjacent to really important coastal wetlands. All these tan areas are coastal wetlands, and they're in many cases right adjacent to very deep water ecosystems, which forms this transition from a shallow coastal environment to a deep water riverine type environment. So it's a really diverse ecosystem. And when I started working here in the 1980s, I'm like, how am I ever going to understand this place? There's 1,800 islands, over 2,000 shoals, and just thousands of hectares of coastal marsh. It's just a remarkable place. There's inland lakes. This is the Lake St. Lawrence. There's like a stream ecosystem called the International Rift that feeds into this very shallow and productive lake of the Isles. There's large embayments like Eel Bay, and then there's the deep water ecosystem where the transoceanic ships travel. And we have a little tiny island. You can almost see it. I think it's right about there. Governor's Island, so it's only a couple of acres, and you're going to learn more about that. But the river has changed dramatically for a couple of obvious reasons, and one was the development of the St. Lawrence Seaway. So this is the emblem for the St. Lawrence Seaway Development Corporation, and it's a waterway that created the opportunity for transoceanic shipping. So it's when the ships are able to go from anywhere in global ports and then access the upper Great Lakes, major ports in Duluth. They created the Seaway through a series of locks and dams. And this project, the St. Lawrence Seaway project, also had a hydropower element. So we have the Long Sault Dam that diverted water around Barnhart Island and shifts it down through the Robert Moses Saunders Power Dam, which is a power dam that's shared by the US and Canada. There's 32 turbines, and they actually have the Ontario Power Generation and the New York Power Authority share the management of that dam. And they also created the Schnell and Eisenhower Lock that allowed for deep draft vessels to access the system, and it was to get around this barrier This is the Long Sault Rapids. You can see that there was a canal that existed for navigation before the creation of the Seaway. But these rapids, there were steamers that would actually shoot these rapids on their way to Montreal. So there's historical accounts of people that would get on these steamers during the Gilded Era in the 1890s, and they would shoot through these rapids. They were really important to the ecology of the ecosystem in the St. Lawrence River. There were major spawning areas for fish, and when this area was dammed to divert that water around Barnhart Island, it created Lake St. Lawrence. Lake St. Lawrence increased in surface area like 300%. So it's kind of like a fluvial reservoir. The water's actually moving, so it's not a stagnant type reservoir. But this is called the South Channel now, which is actually kind of a warm water fishery and ecosystem. So it's changed that dramatically. This is a picture of then and now. And you can see how this is the Long Sault Dam. So these are pretty big changes that occurred physically. There's a fish called the eel that would migrate from Atlantic Ocean to the Great Lakes, Lake Ontario, and up through the St. Lawrence River. And they actually have eel ladders to allow at least the upstream migration portion of that. The downstream migrants still have to go through the dam. So there's issues that NIPA and other agencies are still wrestling with. The part of this story that I'm interested in is the change in the ecological integrity of river systems that are affected by dams. And one of the reasons that is one of my important foci in my research is because there are so many dams in our ecosystems that are interrupting flow regimes, and it prevents access of fish. This is a figure of New York State, and this is from the New York State Dam Safety Database. And there's something around 6,000 dams that are over 6 feet in height in our state. So you can think about the effects on the resources. And a lot of these are small mill dams that were put up historically. Some of them are still generating hydropower, but people built dams throughout our watersheds. And if you look at this picture across the North American continent and also the globe, you see the same story. We've really had big effects on our flowing waters and harnessing them as early sources of power. So understanding how ecological integrity and sustainability is affected by natural dynamic flowing character of water is important. And in the St. Lawrence River, the Thousand Islands section I showed you earlier hasn't really been directly affected physically by the flooding like we see in Lake St. Lawrence. So the physical character of the Thousand Islands, since it's above, the effect of that is not there, but the big effect is through the regulation of flows and levels. So that dam we were just looking at through formally by the Corps of Engineers and through the IJC is managed the levels and flows, and it's actually all of Lake Ontario. So an entire Great Lake and a river all the way down to Quebec City has effects of the management of this one dam. So it's a pretty significant thing. And the IJC I mentioned was formed by treaty in the Boundary Waters Treaty Act of 1909, and they're tasked with an international difficult process of how to manage levels and flows in the system. They recently had the Lake Ontario St. Lawrence River water level study, which we participated in, and it led to the adoption of this Plan 2014, which has been very controversial, and we're going to talk a little bit more about that. But this is a 100-year scenario for water levels at Alexandria Bay, and the red line shows this Plan 1958 DD, which is the plan that was in place until January of last year. And this blue line is the Plan E, so that's basically unregulated. So you're looking at regulated versus unregulated levels, and you can see that there's some periodicity in this system. So before they regulated it, you can see that there's very low periods during periods of low supply that create droughts. And then there's significant high periods. And you can see what's happened in this section after the dam was built, that these water levels are constricted within a narrower band. So this is the big issue. This is what water levels would have looked like without regulation. Some of these are very scary because they're really high water levels, but you also see that there's more variability in the system. So when we dam it, we create a narrower range of flows, and I'll talk more about why that's important. You can also see that in these what we call spaghetti graphs. So each one of these represents a year. So when it's unregulated, you see this great diversity of individual years over an annual cycle. So you get great diversity of flows and levels in that system. And then when we look at regulated levels, you can see it's much more compressed. And when you look at the plan that was in place up until last January, you can see it's really constrained here. They're not allowing the system to fluctuate like this. Plan 2014 still has a high degree of regulation, but it's constrained a little bit less than what we see in the Army Corps of Engineers plan. And they did this because it's the first time the environment was considered in the calculation of levels and flows in the system. So that's just a little introduction onto something I could talk about at great length. But another issue, I mentioned the shipping and the locks. So we have 740-foot vessels that come into the system with 26-foot depth drafts. This is actually a Laker, but there's also Salties. That original picture with the big bow, that's a Salty. And it's created an opportunity for unexpected invasive species to get into the Great Lakes ecosystem that we hear so much about. So right today there's been nearly 190, I think, species, invasive species that have gotten into the Great Lakes system, and the vast majority of these came through ballast water introduction and other introductions associated with the shipping and the St. Lawrence Seaway. So this was one of the granddaddy introductions were the quagga and zebra mussels that are filter feeders. They led to the wipeout of our native unionid mussels, for example. And I'm going to talk more about this creature here, the round goby, and its relationship to viral hemorrhagic septicemia, a virus that's had a big impact on fisheries. We also have a lot of invasive plants, hybrid cattail we're going to talk about. This is European frogbit. It's like a little tiny lily pad-like plant. And then there's a series of predatory zooplankton. One's called Bithotrephes. There's another Cercopagus. This is the bloody red shrimp, and this is Echinogammarus. So there's invertebrates that have also changed the lakes a great deal. And this is the focus of a lot of work and research of many scientists throughout the basin. Oh, that's not supposed to do that. I'm sorry. So I want to shift gears here a little bit and talk about the first researchers who helped found our field station. So back in the 1970s, they had proposals to have winter shipping here in the St. Lawrence River. And it created a great deal of controversy, and they led to a series of studies called the Seaway Winter Navigation Studies. Then a lot of the work happened here at ESF and quite a bit at Cornell. And here's a picture of my advisor and also our former Dean of Research, Neil Ringler, looking at this plastic model of a pike. I don't think they could get their hands on a real one at that time, but this is how we started. They got a Sea Grant project going early on, and it led to hiring of this individual. This is Steve LePan, who is a graduate student and then a research support specialist here at the college that ran the island in the early days. And Steve's in the audience today. Steve, can you hold your hand up so people can see you? There he is. His hair's changed a little bit, but he's still. Steve is now in the DEC, and he works very closely with the Great Lakes, and he runs a field station out of Cape Vincent. And so that early Sea Grant project really was a seed grant that led to significant investment by DEC, and the first major grant was this Federal Aid and Sport Fish Restoration Grant, and it really led to a long-term partnership. Roger Clinton and I were hired on this grant, and we worked on the project. Roger's here as well tonight, back in the 1980s, and it's led to this long-term partnership between DEC, the Fish and Wildlife Service at Hadley, who manages sport fish restoration, and ESF. So that's been going a long time. And it's kind of led to a significant mission. We started with muskies, and now we're working more on a variety of things, but we haven't lost sight of our roots in management of this tremendous species. I'm going to talk more about it at length. But we're here to conserve aquatic resources. We use ecosystem-based science when we can, and monitoring to inform decision makers, because there's people like the IJC, the DEC, the Great Lakes Fisheries Commission. When they relicensed the dam recently, there were over 80 interest groups part of that process. So people need information to make wise decisions about how they affect resources, and that's a big role of our research. And we do this using students, and I have a number of students, present and past, that I have to thank for this, because they're the ones who do a lot of the hard work like you see in these photos. So in a nutshell, everybody remembers the Dr. Seuss book, One Fish, Two Fish, Red Fish, Blue Fish. Well, we do a lot of fish counting in our work, but we do more than that. So, but we're focusing on food web ecology and population ecology of both fish and vertebrates, wetland plants and animals. So we're monitoring all these things in the ecosystem. We have a pretty significant program in restoration ecology, trying to undo some of the wrongs that invasive species and other factors have had in changing habitat for the animals that live in the ecosystem. And we also focus a lot on invasive species. These are some of the principal players. Round goby, which is an introduced fish from the Caspian Sea region that's just transformed the ecosystem, and also a wetland plant, which is an invasive form that's integrated with a native form that's really changed our coastal habitats. So this is kind of our focus, and we have to do this in the context of an ecosystem, and also keeping in mind of climate and anthropogenic changes and flow regime shifts. And also we really use the muskie as a centerpiece for this program, because it's a very charismatic species. It's the top predator in the food chain. So using fisheries, but you can't, the point here is that you can't use fisheries or a specific fish and just study it in a vacuum. You really have to understand its habitat and all the factors that revolve around its ability to reproduce and continue its legacy in the system. So fisheries management is a core part of the process. So back to the diversity of the ecosystem, we have a lot of habitat, we have a lot of species. These are all species we work with. We monitor them, we handle them, we count them, we sample them year after year. The money comes in from managing sport fish, but there's a whole variety of other species that are important in the ecosystem that are important because it is a food web. And so there's probably about 40 or 50 species of fish that we handle on an annual basis, and there's something like 100 to 120 that are logged in this freshwater part of the St. Lawrence River ecosystem. So it's a very big challenge and very diverse. So thinking about the fishery, so why would we want to do this? Well, one reason is it's because it's a big economic driver, and it always has been in this region since people have settled this area. Right now, it's the third largest recreational fishery in New York State, and that's not including Lake Ontario, which is number one, and then Oneida Lake recently took over the St. Lawrence River in popularity, but it's still a major inland fishery. And that's just looking at the US side. So this is an international water, and we manage it jointly. So there's a whole massive part of the fishery on the Canadian side as well. We do have a commercial fishery that it still exists in Ontario. It's a hoop net fishery for what we call rough fish, but that does include northern pike, but yellow perch and other panfish species are caught and sold, and they have been for a long time, many generations in Ontario. The commercial fishery has been closed in New York State. It's really important that it's dependent on natural recruitment and native species. So this isn't like a put and take fishery. This is a fishery that depends on natural recruitment. So that's a very important thing to maintain because it's very costly and difficult to maintain a fishery by the use of hatcheries. So this is a real key factor. It's managed by DEC and the Ministry of Natural Resources and Forestry and through the Great Lakes Fishery Commission, Lake Ontario Committee. And I really want to point out the upper river, with all the challenges it has, one thing it does have to combat all that is this incredible diverse partnerships and conservation with groups, not just the agencies and the college, but also groups like the Thousand Islands Land Trust, Save the River. There's the conservation authorities in Canada. There's the Nature Conservancy, and a lot of like really informed and dedicated citizens that care about the future of their ecosystem. So that's what keeps us going. So now we're going to talk about some of the specific fish we've been working on. One keystone species, if you will, is northern pike. This species is dependent on wet meadow for reproduction. They get quite large in the system, maybe up to 10 or 12 pounds. They've been fished down, so they used to be much larger. And we can see kind of their life cycle here. This is a pike egg, and you can see the backbone and the development of this egg. So they're scatter spawners. They produce a lot of reproductive potential in eggs and can spawn 120,000 eggs per female. And they scatter spawn. They pair up and scatter these over sedge meadows, and they hatch into these sac fry, which will swim up and take a little gulp of air and become neutrally buoyant. And then they feed on zooplankton for a period of about 2 weeks. So the point is you have this little tiny organism. This is about the size of a Crossman BB. Okay, and then it develops into this really large predatory fish, the largest nearshore predator we have. And they have this very sensitive early life that they're demersal. They lay on the bottom and they're at the mercy of the conditions of the habitat. And then when they're young, they're really dependent on what we call lower trophic levels. So they're able to grow and feed, find oxygen to breathe, and find food to grow and develop. So we've really focused on maintaining natural reproduction. So we work a lot on larval fish ecology and early life history. So this is an important concept, is what we call zonation. So along our shorelines, not just in the river, but in all freshwater habitats, and all habitats really have an elevational gradient that interfaces with the water level. And there's my water level going up and down. And this disturbance gradient is what drives the structure of the vegetation. So you'll start out with SAV, submersed aquatic vegetation, floating leaf vegetation. You'll get into shorter emergence and then these robust emergence. Above that, you'll get these wet meadow type wetland habitats. And these are really important for fish spawning seasonally. So my northern pike just went in and they spawned when the water level was accessible. And there are also interactions I've been studying with Dr. Kim Schultz about the release of nutrients from these habitats. So we now know that these coastal wetlands can be 30 to 50 times more productive than the main river. The river has been regulated for nutrients due to the Great Lakes Water Quality Agreement, and they've done a really good job. The water is extremely clear and what we call oligotrophic or sterile, but these wetlands support the bulk of the productivity in the system along this what we call lateral gradient. So when the water level floods them, there's an idea that there's this pulse of nutrients. We've been testing that, and we haven't seen this pulse of nutrients, but what we have seen is that they're very productive, and there's a lot of nutrient that's coming off the watershed to feed the productivity in these systems. So this is just an example of some of the research, and this flooding is what creates a disturbance gradient that creates this diversity in plant communities and animal communities. That stabilization of water levels kind of acts against that process. So that's in a nutshell how that works. So we believe that the infestation of our wetlands with an invasive cattail is related to the stabilization of water levels. So we had a invasion of this non-native form of cattail that hybridized with our very valued Typha latifolia, which is the fatter leaf cattail, and it created what we call hybrid vigor, and it took over every wetland in the system that was exposed to the water levels of the St. Lawrence and Lake Ontario, and it spread landward and also into the system. So these are graphs we use a lot. This is the Thousand Islands Bridge in Cobb Shoal is a bay. Next to the Thousand Islands Bridge that many of you may have crossed in looking at the Thousand Islands. Well, this is one of our long-term research sites, and what we noticed is that when we look at early pictures, before the seaway. All these dark areas are what we call hemi-marsh. There's a lot of interspersion of water, and it's a very diverse-looking habitat. After the cattail invasion really got going in this bay, you can see all these back little coves and nooks and crannies that serve as habitat are being encroached upon by Typha. When we look more recently, it's even exacerbated more. So this is a pretty significant, widespread ecological change associated with an invasive species and the control of water levels that blocks access. This is a tributary creek of spawners that want to reach those wet meadows like northern pike. So there's wet meadows that exist on the back of those marshes that they can't access because of those barriers. So those were pictures. I worked with one of my students, Molly Beelan, and we took a 10-meter peat core. So sediments have been accumulating since the glaciers came through, and we took a core of it. And we did a lot of work on that, but we did a process called palynology where we actually extracted pollen. We worked with Mark Teese in his lab, and we sectioned these cores, we dated them. This was done with lead-210 dating. The deeper sediments were done with carbon-14 dating, and we extracted the pollen and reconstructed the vegetation past in a wetland in great detail. I could show you much more data, but I just pulled out the data that was important to this message. So you can see right here around the peak of cultivation, around the turn of the century, when people converted land, a lot of sediment probably went into the wetlands and nutrient and created a fertile environment for an invasive species just to explode. So this is Typha sparginaceae monads, which were associated with the Typha expansion. One thing we note is that process started before regulation, according to the Corps. So we think that this process was already underway. We just think that the water levels exacerbated it. And you can see the native Typha is this tetrad, and that also increased, but not to the extent of this Typha. And this is the sedge pollen. And we've seen a significant decrease in sedge pollen in the ecosystem over that same time period. So with this long-term perspective, we could really learn a lot more. So that's just an example of the interface of research with our monitoring program. So what does that mean for fish? I thought you were going to talk about big fish and you're talking about plants. But I want to talk a little bit about a study we did. This is one of my graduate studies where I monitored the spawning distribution of marsh. This is Flynn Bay. There's a big Typha-dominated marsh that northern pike would run up and spawn in sedge meadows up in here in Flynn Bay. And we actually did this study in some other areas, Rose Bay and also Point Marguerite Marsh. And we used these egg traps to catch the eggs that these northern pike were distributing over different habitats. And John Forney's here tonight, who is one of my mentors, and I'm really honored that he's here. And when I first started this study, I put out a sampling design of the egg traps based on the literature of where pike spawn. And I met with John, who was on my committee, and he said, "John, why aren't you sampling out here?" And I'm just like, "Well, pike don't spawn out there." And he kind of gave me a funny look. And I added that immediately to my study design after I got that look from such a distinguished scientist. And what I learned was incredible, is that northern pike actually spawn across this entire gradient. And the spawning is in relationship to differences in temperature. The deeper cold water stays very cold. Anybody who swims Memorial Day weekend knows what I'm talking about. And these shallow areas warm up very rapidly. So the pike initially would run up and they would spawn in the sedge meadows, and then they would drop back and spawn in this bay. And then we found them spawning in very deep water, up to 20, 30 feet of water in some cases. And this process, we discovered, has continued in the river since we discovered this back in the late 1980s. And it was driven by temperature. So you can see here the deep cold water, it's less variable. It's much colder in the spring and warmer in the fall. The shallow bay is kind of intermediate. And then the wetland is very warm early. So they take advantage of that productivity that's associated with this early warming that's going to start in a few weeks. And what we found, though, is that the water levels were linked to the degree of deep spawning that occurred. And we've seen this in water levels and also the condition of spawning fish. So when water levels are droughty and low, a lot of fish spawn out deep. They can't access these habitats. And when water levels are really high, like 2017, we see more fish spawning in these shallow habitats. The other factor is they've lost a lot of habitat. So this is kind of unique to large regulated systems where we see this strong gradient. And so we know that protracted spawning has a big influence. So that little meeting with my advisor has led to some significant research related to protracted spawning. We've developed relationships, how it affects genetic structuring in populations. I've had PhD students look at egg quality in spawning across that gradient and develop a new model of the optimal egg and egg nutrients. We've had numerous students, John Cooper and myself, that looked at the timing of spawning and early development, and these things led to the development of a model that went into the Lake Ontario water level study with a colleague, Jerry Mead, and John Cooper and some others. And this became one of the important indicators that was used in the development of a water level management plan. And more recently, my student Erica Augustine has been looking at zooplankton communities and larval forage across that gradient and finding some really interesting results. But the bottom line is that deep and late spawning appears to be a maladaptive process. So this just shows you how research builds. And this next slide, we kind of notice that our habitat, even though there's so much in the river, it's changed. So this is another example. Here's the typha mat, and here's the sedge meadow. No fish can reach that. These things can grow over 20 stems per meter squared. You try walking through it. I found dead pike in the middle of typha mats during spawning runs. So it's a significant impairment throughout all our wetlands. And we kind of thought about ways that we could reestablish connectivity to wet meadow. So the Fish and Wildlife Service stepped in, and we developed this thing called the Fish Habitat Conservation Strategy, working with the Cortland Fish and Wildlife Service, and they have this snowblower on steroids that reopens clogged channels. And I'd like to recognize Scott Schluter in the back, who's here from Fish and Wildlife Service, that helped propel some of this work with Carl Schwartz from the Partners of Fish and Wildlife Program. And I liken it to cardiovascular surgery. So everybody can relate to that. Your heart and all the vessels in your heart get clogged. Well, we reopened those vessels and did some research on that process. And since then, we've done more significant things like creating spawning pools, working with Ducks Unlimited, and Brandy Brown and Sarah Fleming are here tonight, who helped spearhead that project with us. So it's been really exciting to try to restore these landscapes. This is a video of a project in Cranberry Creek outside of Alexandria Bay that was completely choked with cattail other than the main channel. And all these new little back habitats are actually created wetlands that we're now studying. So there's a lot of work like enhancing these spawning pools and connectivity to allow spawning fish to have access, and other animals. I've worked with James Gibbs and graduate students looking at herp fauna and also avifauna in these habitats, and we're learning a great deal about the potential to restore things. Whoa, that's not supposed to happen. I'm sorry. I clicked it once, I promise. Close your eyes. Okay. So moving on. So it's not just limited to those cattail marshes. This is a site where we created a bunch of spawning pools, and we've been studying those. We've also done walleye habitat improvements. This is an artificial riffle that was created, and my postdoc, John Paul LeBlanc, has been studying walleye spawning on this area. This is an example of that channel creation. This is a before and after photo. You can see how there's really no access for fish, and now there's a wet meadow that we located back in here that these fish have access to. And this is an example of a place where people have put barriers across aquatic systems, like those dams I mentioned, and Fish and Wildlife Service and others are creating these bottomless culverts to allow species to exchange. So there's a lot of good work being done, and our job is to study it and see if it's working. I mentioned St. Lawrence River water levels. So anybody who came down from the river knows what I'm talking about, last year's water levels. This is 2017. and the water levels went beyond anyone's expectation. This is my family's cottage outside of Hammond, New York. There's actually a big lawn out here, you know, a patio, and we put up 1,500 sandbags on the island. And so people went through a lot of stress. This is the Clayton Yacht Club. They have a bench on the end of their dock. The dock's underwater. So it was a pretty tough year, and a lot of people are trying to blame Plan 2014 on this high-level water process, but it's really more complicated than that. We had an extreme event in terms of precipitation and inflow to the system that played a big role in these high water levels. And right now, the outflows have been maxed out of the dam as high as they've been historically for many, many months, and they still can't keep up with the inflows coming into the system. So I think this drier weather is maybe going to help them get a hold of that, but we'll see. So is there a silver lining? So did fish respond to the extreme high water levels? So we've been studying that. We do these emigration traps in reference sites and also in our restoration sites, and it created this incredible habitat, flooded habitat for fish like spawning pike. This is a younger year pike that we catch leaving these marshes, and we've been doing this specific survey. Brandy Brown helped me initialize this, but what we see is a really tight coupling between water levels and the production of young fish. And the correlation coefficient of 0.92 is pretty significant in biology, and we see a real tracking. And then during those high water levels, we caught record numbers of northern pike leaving the marshes. So we're hopeful that there might be a strong year class of fish that have been declining in recent years. So the point is that the water levels are really important to the wetland environment, and the data seems to back that up. So I'm going to move on quickly to round goby. Round goby is an invasive species from the Ponto-Caspian region of Eastern Europe that came in through ballast water, and we're really interested in the trade-offs that exist between round goby, their predators, and also native prey fish communities. On one hand, you know, you think it's amazing to get a round goby on the backside of an invasive that really changed the ecosystem in benthic, the bottom of the river habitats, and gobies eat these invasive mussels. They've also increased fish growth incredibly. You see these fish, you don't even know how they can swim. They're beyond the length-weight relationships that we're used to, and they're really incredibly fat, and that you wonder if that health is going to spill over to more natural recruitment. They also buffer predation by double-crested cormorants. Cormorants have shifted their diets toward round goby, which kind of takes pressure off the native fish species that they're having impacts on. And these are all, I'm talking about some very controversial issues, and they've really altered food web balance in the system by bringing that energy that was trapped in the benthos with invasive mussels back up into the top predator system. So we were trapped down here with a system that wasn't producing a lot of fish, and now, you know, gobies are putting on a lot of fish biomass. So the other side of the coin is that they're an egg predator, so they eat a lot of eggs. They displace native fish. They can cause local extirpations. They have effects on angling. Fish get satiated, so they don't want to bite. And you catch gobies all day, so you kind of get tired of that. And they're also probably a vector for VHSV, which is a virus that came into the system. And you're going to see why that's important in a minute, but they're also spreading to other systems. So, and we've seen now they're a big deal in Oneida Lake, and many of the systems throughout the Great Lakes region are also seeing things that we see. So this is a video of gobies attacking a nest of a bass in the St. Lawrence River, and we did studies with my student Christina Calorni to see how long it would take if an angler removed a bass before that nest was destroyed, and some really interesting work. And what we also found is that there's a whole host of native predators that join in on the action, including minnow species and this yellow perch here. So when anglers take a bass off the nest, it really leaves the nest vulnerable, And it kind of depends where that animal builds its nest, the risk it has to goby predation, but it can be significant. You think about a scatter spawning species like northern pike or muskellunge, where there's no guarding male. They just leave the egg there on its own. And you wonder if the gobies would be able to find that, and we've had students work on that problem as well. So this is an example of one of our long-term databases. So we do seining. If people like to wade in water up to your neck, pulling a net day after day, all summer, there's many people in this room that have engaged in this fun process, but we do this in a standardized fashion. We developed a long-term seining index. We do one in July with a 30-foot net and one in August with a 60-foot net. This data actually was standardized, and you'll see some more of it in 1997. So we've had 21 years of continuous standardized sampling. This I've learned from my work at the Cornell Biological Field Station about the power of long-term data in understanding ecosystem change. And I helped develop long-term data sets at Tibbs, and we've kept them going, and they've really helped us bring in funding and understand what's going on. This is one example. So this is the tessellated darter, Ethiostoma olmstedii, which is a small benthic native fish. Muskies like to eat them. Baby muskies eat these. It used to be a pretty big part of their diet. And now round gobies have invaded these sites. And this is our tessellated darter counts in our seines. You can see these are pretty big numbers. And then gobies, including our trap net survey and 2 different seining surveys, have increased dramatically. And 2017 was the first year in all this sampling we didn't catch a tessellated darter in our series. So one of these data points represents 90 seine halls. So think about the effort. The students, it's a crew of 4 or 5 people go out and collect this data, and they're doing that over 20 years in 11 different bay and wetland complexes throughout the river. So and this happens throughout the Great Lakes Basin. There's a lot of dedicated people to collect the information we need. I'm going to move on to muskies, because this is the big highlight of the talk. Muskies are a top predator. They made Gordon Grice's book of dangerous animals. So they're listed in Deadly Kingdom if you want to read some trash reading. But this is you can see why they made it. This is actually the jaw of muskellunge. You can see these large teeth, and then they have these thousands of what are called villiform teeth. And they're designed so if a fish swims in there, it's not getting out. So that's the whole idea. They're curved backwards. And this is the native range of muskellunge. So this is a North American endemic. It only exists here. So it's kind of a special animal because it's really large. It's endemic. We have worked a lot on the genetics, and we've defined the genetics throughout the Great Lakes and other drainages in recent work, teaming up with other scientists. It has a great cultural value, especially in Clayton, and a big trophy fishery. People have been introducing these outside their range because they're so popular, but it's created some significant controversy. And we really work to focus on the native range. The life history of muskellunge is similar to pike. They have an egg that's just a little bit bigger. That's the same fry, I know, as the other picture, but they have a very similar zooplanktivorous phase, yolk sac phase. This is actually a wild fry in some emergent plants. And then the juveniles are just absolutely beautiful. They're this golden yellow, and they have this yellow-gold stripe that allows them to hide in vegetation, especially water celery, which is Vallisneria, so they can hide in there, and they have this cryptic coloration, which may help prevent avian predation. And then they emigrate from these spawning and nursery bays that are so important to their life history and as subadults. And we know very little about this stage of subadults. But we do know that the adults have significant spawning site fidelity to the sites that they'll stick to the same spawning sites year after year. So there's an example. There was a fish that I tagged and I caught 19 years later. I was really excited on one hand. On the other hand, it's like, oh my gosh, I've been working here a long time. But the fish we caught 3 or 4 times, and it came into the same embayment to spawn. And it's just a really unique life history. We don't know if this is natal homing, but it's suspected that it is from other studies. And you can see this is one of the seining crews releasing a young of the year muskie into a nice habitat. I think this is a juvenile muskie, and you can see that stripe that allows it to just merge right into that habitat and have that cryptic coloration. So it's this natural reproduction that makes the river such a special place. Many populations are sustained by stocking, and the river has this real ability to produce young muskies. So that's what we're trying to protect. But exploitation's a big part of the picture. They have this legacy as a trophy fishery. So this is a famous event on Leech Lake where the muskies went berserk, and they don't know how many muskies they caught, but they took a lot out of the lake in about a week's time. And similar things happened in Clayton. It was a boom fishery, and it's probably because of this. These are the competing world records that existed at the time. Art Lawton caught this fish that was apparently 69 pounds, 15 ounces. So this is our state record. It was the world record for many years, and he ate that fish. So there was no real record other than these pictures and signed affidavit. And then, Wisconsin, they want the record, and Ontario wants the record. So there's this big controversy that I'm not that interested in. But what I am interested in is how it brought attention to the Thousand Islands ecosystem. And they actually started a derby, and the derby was a muskie derby, and all these fish were killed and taken out of the ecosystem. And the derby got worse and worse and worse until they canceled it in 1978. And that's kind of the time when people were worried about we're going to lose this resource, and they contacted DEC and ESF and a lot of intense meetings. But you can see some of these fish that were being taken are really small, and the numbers were taken out in pretty big numbers. So it led to the development of a muskie management plan, and it was to perpetuate muskie as a viable self-sustaining component of the fish community of the river and to provide a trophy fishery. So first as a part of the community, second as a fishery. This is Jake Runner, one of my lab managers, and Brandy holding a really nice muskie. And this ended up in the New York State Fishing Guide, so it's really exciting for us. And we also got a special issue, our cover photo, a year and a half ago in the magazine Fisheries that features our program, and also there's a special issue on the management of these fishes that was co-written by me but led by one of my graduate students. So really exciting. Internationally managed. This is the basis for how we manage them. So what's ironic is this bone is a Clythra bone, and it comes from the pectoral girdle of a muskellunge. So the bone that the gill cover rests on when it's breathing is the Clythrum bone. And you can't get this without killing the fish. But it's the only accurate way to age the fish, which is crucial to management. So that's kind of a conundrum. So Steve LePan and Dr. Werner and myself, we started programs working with taxidermists. So we had to work with the people taking the muskies to get this data. We also worked with John Castleman, who is famous in Ontario for muskie management, and used this bone, built a project on it. But what you can do is you can compare the length of a fish. This is fish 101. So everybody pay attention here. These are females and males. This is age in years. So you age these fish, and you look for these annuli, so they cease growth in the winter and re-begin their growth. So the growth cessation creates this dark mark, and you can age fish, you can reconstruct their growth rates. It's an amazing tool in fisheries. You can calculate growth curves. I won't get into this guy, von Bertalanffy, but you have these curves, and then what they allow you to do is compare your size limit and see what portion of the population you're protecting before they spawn. They spawn at about 6 to 8 years. So if you have a size limit of 36 inches, all these fish are being protected, but they're not even reproductively mature, and they're leaving your population. So that could influence your natural recruitment. So we've seen a progressive increase in size limits that have allowed fish to spawn more and more before they're available for exploitation. So that's a basis to it. The history of regulation shows like hardly any regulations for many, many years, and then increasing sizes, reducing bag limits, increasing sizes to 54 inches today. And it's because of that relationship. But more importantly, community groups like Save the River have promoted the release of muskellunge, legal-sized muskellunge, and now all muskellunge over 48 inches. And they get this print that was done by a local artist. There's 5 renditions of this print now, and it's promoted a catch and release philosophy from that take everything philosophy we had historically. We've seen the size limit increases. We're identifying critical habitats and monitoring the data. So all the pieces are coming for a really impressive management. So, and this is where it works. It doesn't work unless your anglers buy into it. And initially, we didn't have the trust of the local community. We've built that trust over time. This is a guide, Richie Clark, who's worked with us a long time. There's probably other guides in the room. I tip my hat to every one. And he has this statement. He caught this 60-inch muskie, which is a potential world-class fish. And here's what he said: if I didn't think it was going to live, it would be on my wall. But I live on the river, and when you take a fish that big, you take a piece of the river, and I didn't want to do that. So we've seen like almost 100% compliance of people releasing all the fish they catch to maintain this population. They depend on it. They know the river depends on it, and it's been an overwhelming response. So we've identified all these habitats using early radiotelemetry studies, field studies, a lot of seining, thousands and thousands of seine hauls in the standardized index. And now we're using hydroacoustic arrays, working with Steve Cook and other groups at Carleton University, where these mini tags go in the fish, and we're learning more about subadult habitat use. And we've identified all these places that hopefully will be protected for future recruitment and generations of people to enjoy. So did it work? We can do a lot of hand waving, and but it's really important to do the hard work to understand if there's been a response. And we did that. We looked at size distributions of muskies before and after voluntary catch and release, and also size limit increases from the 80s to the 90s and then up to 2000. And we saw this amazing modal shift in size of many bigger females and males in the system. So this is evidence of fishing down. When people take out large fish, they cause the size distribution to shrink. So it's called fishing down. It's a classic response to overexploitation. When you release the overexploitation, recruitment and the fishery can recover. And we saw evidence of that, more young coming into the system. And then the worst possible thing happened. An invasive species came in, actually an invasive pathogen, viral hemorrhagic septicemia, and we started counting carcasses floating all up and down the system after all that hard work of conservation. So there's this rhabdovirus. It's a virus that came in 2005. Evidence it occurred in the system in 2003. It was confirmed in muskellunge. My collaborator here is Rod Getchell from the Cornell Fish Health Laboratory, and Paul Bowser. They worked a great deal on this. And we've developed tools. We've looked at temporal progression of prevalence in smallmouth bass. Jeff Eckerlin, one of my former students, and we found that muskellunge are highly sensitive. There's a couple of different studies that have been done on that. There's many fish affected by VHSV, but none more than muskellunge. And there's the isolates of this virus are changing too over time. There's something like 190 to 100 new isolates of the virus, so it mutates and changes. And we see that there's a linkage to round goby. So it's really a remarkable ecosystem change. And this is what's happened to our spawning sites. We have long-term index in some of our spawning sites. You can see the catch in our nets of spawning muskellunge that we monitor. We tag fish, and we could see a decline. And then after VHSV, some of these sites no longer produce muskellunge, or they're severely reduced from what we saw historically. The kicker is we monitor reproduction of young muskellunge at 11 sites. Like I mentioned, each one of these data points is 90 seine hauls. Here's data from 96 to 2017. VHSV came in the system along with round goby, and we've seen a real collapse in the number of young that we see on our nursery grounds. These are the cruise out seining. So we got a real problem here that we're dealing with now. And we also maintain an angler diary program. So all these guides and ardent anglers monitor their catch and the number of hours they fish. This is the effort line, so it's very variable, but over time it's quite stable. This is the catch line. So our management goal is to have 10 fish, or 1 fish per 10 hours angled, and now we're way down here. So the management was really close and above our goal for a period of time, and now the fishery has taken a severe twist. One of the silver linings is the fish that are being caught are absolutely enormous. So people are seeing these really big muskellunge. You can see them in these pictures right there and there. So the fish that remain are incredibly big. There we go. So we're working with DEC and our other partners to try to see if the habitat can still support muskellunge. So we've been raising muskellunge in our lab at the island. We actually marked them with a chemical marker so you can see that they came from our lab, from their otolith bone. So we sacrificed these as young, a sample of the fish that are released. These are this is a fry stocking program, and we release these fry in numerous sites, and we can tell a wild fish from a caught fish by transmitting UV light through this otolith bone, which is an ear bone of the muskellunge, to tell it came from our lab. This is a baby muskellunge that's being fed Artemia in our lab, and it's a lot of work to raise these fish and mark them and study their survival. So, and we're working on a new project now, releasing larger fish. Last year we released 4,500 young of the year. I tip my hat to J.P. LeBlanc and the rest of the TIB staff for raising these fish in our lab and tagging each one of them. And we're focusing on restoration of lost spawning and nursery sites. We use local broodstock and genetic pairing to get genetic diversity in these fish. And I have a graduate student, Kate Abbott, who's here, who's studying survival and distributional studies of these fish, and also how are they going to react when they're exposed to VHSV. So there's a lot of detailed study that goes to this, and one of the unique parts is she's looking at the immune response. So hopefully these fish, when they get a low-level exposure, will develop some immunity to the virus that will protect them in the future. So in summary of that story is that we know that water levels and flows and temperature are really master variables that drive recruitment. There's a lot of research that we've done to look at this. We know that invasive organisms can alter community structure and also create a lot of uncertainty in your management. It used to be, just managing fisheries, and now it's a whole lot more. Native species still hold top predatory roles in the fishery. So this is a bright spot. With my student, Brent Murray, we studied resistance to ecosystem change in the population. Native species still hold these roles, and they still exist, and they're recruiting. So we know that protection, restoration, and management are the keys to future recruitment. We know that long-term data coupled with targeted research is going to help us understand and face environmental problems. And we depend on collaborations in our community, the commitment of people, and our funding resources to keep this project going for so many years. So I just wanted to move from that and show a little picture. This is our current group of staff and graduate students that are working on various projects that you may be hearing from in the future. It's also very hopeful the next generation of young scientists that are equally committed to this resource. There's been so much collaborative involvement at ESF with all these faculty, some really interesting studies, and then groups outside of ESF that we collaborate with. A lot of great work, so I just wanted to show them just to make sure they aren't forgotten. And also past graduate students, so many of you might see your name on here. Dr. Werner started a lot of this. James Marin worked on the project in 1976. So that was the first graduate student, and you can see this long line of students that have graduated from ESF that have been a part of this immense effort that you're just getting a flavor for. Also the past staff and students, lab managers, we really need to have an alumni event at TIBS because I think there's a couple hundred or more technicians that have participated in the program over the years now. And Governor's Island, it's just a fabulous little field station now, and it was founded, the name Governor's Island came from Thomas Elvert, who was the Lieutenant Governor of New York State, and I recently found out that he was the Syracuse native and was on the Syracuse legislature for many, many years. And they named him Old Salt because he developed the salt springs here in Syracuse. And he lived 17 summers on the island. So he loved this island. I've found writings he did recently. Jim and Tony Lewis are the people who donated the island to the college. They were great benefactors to the Antique Boat Museum, the Clayton community. Used to be called the Ellis International Laboratory, and that was her maiden name. And I did get to meet them several times. And at that time, we just had a couple buildings and an old boathouse that we made things work, because that's what we do. And I just wanted to finish with this little video that kind of shows last summer. The internet's a little slow. There we go. So this is a beautiful summer day on the island last summer. For many of you who have worked there, we're flying over the island here with a drone. All these buildings have been renovated. I want to thank Physical Plant for their help over the years in helping us maintain this site. But the students do a lot of the work. They help us open up and close down. And they've helped. This is our new building that was done through a massive fundraising effort. And I want to thank Brenda Greenfield for her help, and many others, Bob Quinn, for helping us raise the funds to create a new dormitory and staff facility. We also received an NSF grant. I want to thank Kim Schultz and Neil Ringler and others for that NSF grant that we all partnered on. That led to the rehab of our entire lab. So it's just a wonderful place to work. This is the yacht club where we keep our cars, and we have a new mainland facility there as well. So just a wonderful group of committed people that are helping that little island make a big difference. So thank you. Appreciate your time.
So this talk is open for questions, and as a reminder, we have a microphone over here in the corner. If anyone, would like to ask any questions, please don't be shy. And again, if you have trouble to get over there, just raise your hand and we can have a microphone come to you.
No questions.
There was a lot of information there.
Yeah.
I'll start with a question, just to try to get things going here. And in that, what you've presented here is a huge amount of work and a huge amount of partnerships. So how do you manage something this big, right? And still have time to teach and sort of mentor our students? This is a phenomenal job, in my opinion.
That just reminded me, I want to thank my wife. Because it's been a big challenge, but I'll tell you, I worked as an intern at the Cornell Field Station, and that kind of built a model for me of what I could see on the river. And so we've kind of followed that. And, we have so much help that it's, all the young people at ESF get this great opportunity to work at the station, and it's just a big collective effort. And I want to thank the college and its administration for supporting this, because in the early days it might have seemed like maybe a crazy bet, but it's really paid off. So it's it's been a collective effort, and we just are all in, and that's how we do it. So.
What do you think the future of the muskellunge population is going to be up there with PHS?
Good question. So Dr. Bob Werner, one of my mentors, is asked what's the future of the population. The recent paper we published in the book is one of uncertainty, which is, hits me. So the muskie is resilient. It's been there since post-glacial time, since it came back from Mississippi Refugium. So it's I want to think that it's going to be part of this ecosystem for a long time. But some of these invasive species and changes they took a pretty hard hit there. What's interesting is Lake St. Clair also had VHSV, which is another large lake in the Great Lakes that hosts a big muskie population, and they lost thousands. But that fishery is doing better than ever. For some reason, Lake St. Clair has bounced back and the St. Lawrence has struggled a bit. But we have a lot of habitat, a lot of hard people working on it. If the muskies can adapt to the changes in the virus over time, I think they'll be just fine. Yeah, Maggie?
So you mentioned that muskellunge are really susceptible to VHSV. Is there any sort of like resistance breeding program, so breeding fish that are more resistant, selecting for that?
Yeah, so Maggie asked if there's any programs to breed resistance into the population. It's kind of some of the research that we're getting into is looking at the immunology that Kate's going to be working on, and we have partners in Rod Getchell. Chris Whips is also working as part of our team, and we're going to develop tools to look at how muskies respond from an immunological perspective to exposure. When you start to breed animals purposefully, we've had somewhat of a sordid history with that, and this is a native population. We just did a genetic study across the entire basin with a bunch of my colleagues and found that significant amounts of native muskellunge diversity still exist across their range, which is amazing considering what we've done to them. Okay, so you think of people moving strains, moving fish around, destroying fish populations, creating other ones. The bulk of the muskellunge genome is still intact. So we really think that those natural processes that drive evolutionary forces are the ones to help us get through this. So, Neil?
Did the gobies bring the VHS?
That's not known. Rod's going like that. They came at the same time, and I kind of glossed over the goby problem because there's potential that the goby and VHSV are like a 1-2 punch because the goby are changing the whole fish community and the environment that the muskie lives, especially in that sensitive stage. And we know that muskie in controlled conditions gobies can eat muskie eggs pretty fast, and they can find them. So, that's another issue. But I don't know if you noticed in that data with the darter, we've seen a big decline in goby last year, and I don't know if that's due to the high water levels influencing their distribution or our gear, or if we're starting to see a decline in goby populations in the system. So that's where the monitoring comes in.
Just as a reminder, if you have a question, could you please, because we're live casting this on YouTube, if you could come over to this microphone, it would be really helpful because then the people who are not in our current studio audience will be able to hear. So I apologize for that inconvenience, but it'll be helpful for some of the people who are viewing this online who couldn't be here today.
I was just wondering if you have any problem with lamprey eels?
Lamprey eels. So yeah, lamprey eel is a species in the system, but they're managed intensively by the Great Lakes Fishery Commission. So they keep close tabs on them. I have seen lamprey eel attacks and have seen them attached to muskellunge. Lake trout were extremely vulnerable, and they crashed the lake trout population in Lake Ontario. And since the management efforts by GLFC and so many people, they've been able to suppress the populations to allow wounding rates to decline substantially. Thank you.
So we're gonna move on to the next stage because we actually do have some more things here for John, and maybe he's not completely aware of it. But traditionally for the Travis Lecture, Dale Travis is one of our many distinguished alumni from ESF here. And he's a graduate of Wood Products Engineering in the class of '59. And in the tradition of the Travis Lecture, we always give our speaker a gift showing some woodwork. So I'd like to invite John back up to the stage to give him this gift.
Oh, wow. Very nice. Yeah. Beautiful bowl and a cutting board.
So,
Thank you.
so these are actually made with wood from ESF's forestry program. So this is maple from our own forest. And in addition to this, as you've seen already, John has done an incredible amount of work. And for this work, the college has also recognized John as the ESF exemplary researcher. And so I'd like to present him with a plaque for all of his great contributions that he's made for the college and for the state of New York and fisheries, basically worldwide, with the work that he's done.
Yeah, thank you, Chris. Thank you. Thank you.
So I'm sure John is going to stick around. We have a great reception outside that I'd like to invite all of you to come out and attend, and I'm sure John will be happy to answer more questions. But let's all give John another round of applause. And finally, once again, I'd like to give a round of applause to Dale Travis for making this whole lecture series possible, and to thank you all for your great attention to this event. Thank you very much. Yeah, let's go get some stuff outside. John.